So the data that our group is presenting here at HIA 2022 is based on a combination that we've been exploring in the lab of Tolinapant, that is a potent antagonist of inhibitor of apoptosis protein, and the hypomethylating agents Decitabine. And we have basically explored the rationale of combining these two drugs in vitro in our preclinical model of T-cell lymphoma. So in a large panel of T-cell lymphoma lines, we have shown first that Tolinapant has different activity depending on the cell lines, and then we have explored the synergistic interaction in between Tolinapant and a number of agents that have some activity in peripheral T-cell lymphoma that are already in the clinic. And we have found that Tolinapant is particularly and strongly synergistic with the hypomethylating agent Decitabine. So in the poster, we also show that potentially Tolinapant acts and induces cell death via the activation of apoptosis, but also via the activations of the necroptosis pathway and therefore immunogenic cell death. So in the abstract, we show that potentially the rationale of synergistic interaction between these two drugs is based on activation and further enhancement of the necroptosis pathways. So these data are interesting, they're new, and they suggest how this combination of agents could have a role really in patients with peripheral T-cell lymphoma that are generally very chemo-resistant and they don't have a good outcome with the standard chemotherapy. So there is a phase 1-2 clinical trials that is already on the way, and these preclinical data support the use and the exploration of this combo in patients.